I am Kailing, the Regional Field Application Scientist of Zuba Systems, and today we will be looking at Sapphire FL Biomolecular Imager, looking at it from the perspective of how it helps in terms of small model organism imaging, or precisely, we will looking at how it helps in monitoring in vivo tumor progressions by looking at generating these such data like tumor size and monitoring the changes over time. So before we go into the application side of this, let me kind of give you a brief introduction about Azuba Systems. So Azuba System is a United States-based company. We are Western blot solution provider. We provide cell systems, reagents, and consumable for you to perform your Western blot, covering both chemi and fluorescence Western blot, and also imaging system to capture the pictures of your Western blot as well. So here's our company portfolio. There are several key instruments in the company portfolio. The one we're going to focus is Sapphire FL Biomolecular Imager, which we launched in this last year. Sorry, last year, 2023. Okay, right. So it is a next-gen fluorescence laser-based scanning system. Okay, this is our highest spec equipment, and here we expand its application and venture into the world of in vivo animal imaging. So, give you a general idea about the imaging solutions available in Azuba systems. So, we have three equipment that can basically cover up your routine laboratory work. We have Azure imaging systems, Solo, and also Sapphire FL. Okay. So these are the applications that you can be able to generate, okay, from these three instruments together. Okay. So Azure imaging systems will be the one that you usually use in the very generic application like your gel agar gel electrophoresis, SDS page, and SDS page including gel and also Western blotting as well. Okay, so it covers all the way up to chemi and fluorescence imaging. And chemi solo is very simple. It is dedicated for chemiluminescent signal imaging only, and some very minor protein gel imaging. But the star today is Sapphire FL Biomolecular Imager. Okay, so you can see there's a huge difference on the way we jump from Azure imaging systems and ChemiSolo. So we're moving on to fluorescence image or fluorescence signals capturing, but in a very high resolutions and very highly sensitive as well. So this piece of equipment aim to give you the sensitivity and also the low background noises that you need, okay, in terms of quantifying the data either from your microarray, plate-based assay, or animal imaging, which we will be talking about later. And then, of course, on top of that, this piece of equipment, Sapphire FL, can also do phosphor imaging on your radioisotope label samples. Okay, so Sapphire FL. If you look at this is how it looks like. Some of you may have already visited or see this or saw this in KSBMB recently. Okay, so this is how it actually looks like. Okay, so in terms of the optical module or the fluorescent channels that we talk about in Sapphire FL, it is an uncoupled optical module design where you can mix and match or switch around your filters and laser combinations. At this moment, we have 12 standard optical modules allow you to choose from, okay, which we paired the excitation and emission already. So you can see it covers up from your UV range 488, okay, 532 sci-fi, sci 2, sci 3, sci 5, and sci 5.5, including the phosphor imaging as well. Okay, so Sapphire FL, if you look at the piece of equipment itself, it has a very large overview, okay, 25 times 25 cm field of view, okay, and then we have a 4 centimeter of clearance, allowing you to image samples with different height or we say different depths. Okay. And then with this piece of equipment, you can achieve high resolution imaging up to 5 microns. So this will be very useful for glass slide imaging or 364 well plate imaging as well. Okay. Or microarray. Okay. And with that 4 centimeter clearance, it comes together with a Z plane adjustment for your focal planes. That means you can adjust the focus of the scanning at different depth. Okay, we covered from -1 up to +6 mm above the glass surface. Okay, so this allows us to image a wide variety of a sample. Okay, then with additional 5 gaseous anesthesia port, this will allow you, or researcher, to do in vivo live animal imaging, or specifically rodent imaging like mouse and rats. Okay, right. So in if you look at how we apply the different Z-plane or different focus allow us to image different samples. So for example, if you want to image your plate, okay, fix cells on your plate, a cell culture plate with a very optical clear bottom, then the focus will set at 3 mm. So if you want to image something like a membrane, okay, like your PVDF membrane, okay, that is flat on the surface, that we will use focus at 0 mm. So on the side of the in vivo imaging, this is the part where we will utilize the Z plane scanning. That means you can start off by scanning at 0 mm, 1, 2, 3, all the way up to 6 mm deep into the tissue, okay, or deep into the body of the sample that you want to look at. So this not only covers in live animal like rodent, mouse models, it can cover for C. elegans, zebrafish, or any kind of uh, organism that you want to see their signal in depth. Okay, so this is how we utilize the different focuses that we can use in Sapphire FL. So it is very important to optimize the focal plane because there you will see all the signals at different depth. With this, I'll bring on to the confocal imaging technology we have in the Sapphire FL. Okay, so it's very quite important to optimize the focal plane when you image sample with depth, for example, like a living mouse. Okay, so this such important step, okay, is because of the confocal nature of the Sapphire FL. It uses PMT and APD detector. Two of these are the best detector for fluorescence signals detection and also the phosphor imaging as well. So each of their different wavelength. Okay. So most importantly, the pinhole aperture of the confocal, you can see there's a pinhole here that allows you to scan, okay, the detection of only the light coming from the focal plane. Okay, so anything out of the focal plane, like above and below, will be eliminated. They will not go through. Okay, they will, only the one at the focal plane will go through the pinhole, reaching the detector itself. So, what this does is it enhances the resolutions, okay, and also eliminates a lot of background noises that you might actually worry that you see in the depth scanning. Okay, so you eliminate out of focus light from above and below the focal plane, and that's why these will enhance resolution in brief and the contrast that you will obtain in the images. Okay, right. So all these images that we scan at different focal plane allow us to take a Z scanning, okay, Z scanning on the images, okay, so because each of them will have different signals at different depths, we can either make a merge image or showing them in the sequence or a GIF file to show the differences in the signal at different focus. Okay, right. So next thing is key features in Sapphire FL. Okay, this Sapphire FL is highly sensitive. As I mentioned before, it uses PMT and APT detectors. Okay, and we have the customizable and user changeable optical module that you can swap, okay, mix and match the laser and the filter combinations. All right, and then what is important or what are important for the in vivo animal imaging will be these three features that I mentioned before. Firstly, the wide adjustable Z plane, which is a focus plane that we allow to do up to 6 mm. Okay, then that at that focal plane, we can decide what kind of resolutions that you want to go for. You want to go for 5 micron resolutions, okay, or 200 micron resolutions to see the general, the image of the tumor also. Okay, that will be up to your choices and also the requirement of your data analysis. And most importantly, Sapphire FL incorporated 5 gaseous anesthesia ports to image live animals. Okay, so those that who needed gaseous anesthesia, for example, like mouse and rats, that can fit into the 4 cm clearance, height clearance, that will be available to scan in Sapphire FL. And Sapphire FL also comes with a Cami module if you needed this capturing as well. Right. So let us move on to imaging the living small animals then. So Sapphire FL is designed to allow imaging of the living small animals under anesthesia. That's why it has the gaseous anesthesia port. Okay, so it has a 5 built-in anesthesia port for to image multiple animals at once with 4 cm clearance between the glass imaging surface and the lid. So you have plenty of area and also the height for you to fit in for small rodent. So in general, Sapphire FL allows you to generate in vivo assay data, okay, which in a faster and more consistent manner because you scan them in one go, okay. So everything is together and keeping it very time and labor efficient without sacrificing too much animal. So sacrificing part is about the time point analysis of the in vivo imaging. Okay, so if the certain experiment that requires a full length of observations or monitoring of, let's say, a tumor growth or tracking of the compound, there they don't need to sacrifice or make up so many experimental group for time point or tracking analysis. Okay, but some will require that for control as well. But most importantly, this feature allows Sapphire FL able to generate data for health-related research. For example, your cell migration studies, preclinical drug testings, and also tracking of drug or medicine or certain treatment over the time in the animal model. Okay, right. So this is something that in situ assay or Western blot, the other things that couldn't provide because this involves in tracking and monitoring changes that you can visualize it in a single image. Okay, right. So while so many studies require these dedicated, if you want to do these kind of studies, okay, you do need dedicated small animal imaging equipment. For example, like they are quite normally are quite expensive, very costly, and they are bulky as well. And many labs are not having the direct access to that, okay, because they either need to go for the to call facility or it is not really available in the animal facility itself. So, what Sapphire FL able to provide is the ability to quickly gain these prelim or quantitative data from a benchtop imager. So, it will drastically shorten the time for your confirmation or even data analysis as well. Okay, right. So next is let us look at the application note that we're going to talk about later, which is an in vivo imaging and monitoring of the red fluorescence protein plus or positive tumors in the mice using the Sapphire FL. So these involve imaging of living animals under the anesthesia, and also it also involve image of the excised or harvested tumor or tissue to compare or tally the results that we see in the living animal itself before we sacrifice them. Okay, so monitoring tumor progression in mice, this is a very powerful approach to visualize a structure labeled with markers or dyes. We know that in all visualization method that we use in research or R&D purposes, okay, or diagnostic and anything, so visualization is always the key. So we want to look at, let's say, DNA, protein expressions. We want to look at them, but we aren't able to do that. So we need something to tag with it. So this application or this concept applies the same thing for anything you want to see or visualize within the animal itself. So in this case, it will be the ability to image tumor in vivo, which is in the animal body itself. So this will generate tumor size data, allowing you to monitor the changes in tumor size over the time. So without sacrificing the animals. So you don't need to do time point grouping like the one they sacrifice on day 4 or the one that you sacrifice on day 7. You don't need to have so much animal to sacrifice. So you can monitor them over time and maybe sacrifice a representative for that time point just to tally the data. Okay, right. So all in all, it allowing you to study the tumor progression and of course the most importantly the effectiveness of any anti-tumor or anti-cancer treatment or drug. So this is about tumor progressions. Okay, it applies the same thing for tumor initiations or any kind of anti-cancer studies. All right. So what Sapphire FL play in such a role is it offers you or researcher a benchtop in vivo imaging options. So while all these things can be easily done, okay, on a benchtop, and it can be easily incorporated in your routine workflow without going back and forth to a call facility or booking the bulky instruments and so on. So these serve as that purpose, right? So let us look at the details on how to image or monitoring the red fluorescent protein positive tumor. Okay, right. So, firstly, the cancer model. So, we need to look at what are the models that we're trying to set up right now. So, the cancer model is a human breast cancer model. It's an animal model that resembles the stage 4 human breast cancer. That's why the injection of a 41 tumor cell, which is a mouse mammary gland tumor, is injected into a 6 weeks old female biopsy mice. Okay, so this is a biopsy mice used in uh, this research or application. Okay, in your research, you might actually want to consider about new mice, which will actually extremely helps in terms of imaging and visualization of the signals, okay, without the fur autofluorescence signals in a disruption, right? So there are two experimental group here. So in one group we have two mice, which they are control group. We have RFP negative tumor, okay, because they are injected with just the wild type 41 cells. So they are lacking the red fluorescence protein reporter. Okay, that is the control group. For us to look at whether the tumors are growing, and then if there is tumor, do we see the signal or not? So these serve as, I will say, is a reporter control, okay, or a signal control. And we have another group of experimental mice group consists of three mice, okay? They have these injected with 41 cells with red fluorescence protein tag and stable expressing the fluorescence reporter, which is turbo RFP. okay, a turbo red fluorescence protein. So we know that if there is a tumor, we will see the red fluorescence protein in the mice itself. Monitoring parameters are listed below. So the injection site is actually scanned on day zero, but the one that you are useful for tracking over time will be the subsequent days or day, depending on the growth of the tumor itself. For this case, we take day 4 and day 11, okay? And then one of each actually sacrifice on day, uh, two of them basically, day 11. So we harvest them to image the tumor ex vivo, which means after we harvest it, and then we put it on the Sapphire FL and scan it. Okay, just want to tally the data that you scan before you sacrifice them. All right, so that is the purpose of ex vivo scanning. All right. And the fifth mouse is sacrificed on day 14. Same thing. After we scan on day 14, we sacrifice on the tumor and then harvest the tumor and scan ex vivoli. All right. And the tumor length and width are measured by external caliber. Okay. And then the volume is calculated based on the modified episodal formulas. So I'm not going to mention the formula here. So basically, the formula is listed over here. It's a volume equal to blah blah blah. Okay. Right. Next thing, in vivo tumor imaging. So, importantly, the tumor site or the injection site must be shaped because we want to eliminate the background signals of autophorous signals from the fur itself. That's why it's highly recommended to do it on nude mice, okay, to ensure that kind if you want to get more control over the signals. So we used 2% isoflurane, okay, with oxygen flow rate set to actually it's a 2 liter. I'm missing the two there per minute. Okay, so all the in vivo images, the capturing at the site of the whole animal were captured at 100 micron resolutions, and then using the 532 standard optical module, which is your side 3, okay, side 3 channels, okay, with intensity 6, and the focus actually set starting at 0. So we set at 0 and then all the way up. Okay. So it's always recommended that if you want to image a live animal when they're under gaseous anesthesia, you shouldn't exceed more than 1 hour because if possible, we want to limit it within 30 minutes of scanning. So this is very important because you do not want them to lose their body heat, okay, to that might actually change the biology, not biology, but the metabolism, or actually make a changes on your subject, okay, on the animal subject. So heating should be provided for long-term imaging, and then you do need to have a proper nose cone to provide the isoflurane to keep them sleep throughout the entire scanning itself. All right. So that is the recommendation that we suggest for anyone who want to do live animal imaging. Okay. Right. So ex vivo tumor imaging, okay. At this point, the tumor will harvested after we sacrifice the mice. They capture at 50 micro resolutions because we want to look at the details of the tumor itself, and also using the same the 532 standard optical module that is for your red fluorescence protein. All right. Okay, so here, this is what I mentioned. You have 4 cm of clearance. Okay, so anything that fit within 4 cm, for me, this is will be the thickness of the mice, the biopsy mice. It fits within, is definitely fit. Okay, then you can image them, and then we have about 5 gaseous anesthesia pot for multiple animal imaging at once. Okay, and in terms of result, and this is the length, the width, and also the volume that calculates based on the formula modified formula that I mentioned before. Okay, so this is a tumor measurement progression from day 4 to day 11. Okay, right. So next is let us look at the in vivo tumor imaging on the biopsy mice. Okay, well, so while Sapphire FL is not really a dedicated animal imager, so it can capture publishable images of the fluorescence tumor, as you can see over here. They are in vivo fluorescence tumors image. So in this study, they mentioned before, the mice were injected with red fluorescence protein expressing 41 cells, which is a breast cancer cell from the mouse. Okay, it's a animal model for stage 4 human breast cancer, and we image after 11 days. So this is day 11 image, all right? So we can see the growth of the tumor cells. So you can see they are also the autophagy signals from the mouse fur surrounding the core of the tumor itself. Okay, so we need to shade off the fur at this region, so ensure that just to remove as much background or the autophagy signals as much as possible. So we want to look at the tumor itself. Okay, right. So the image at the center show that this is a torso area, okay, of the mice under anesthesia with the red fluorescent protein positive tumor in the middle. So the tumor itself is able to be highlighted in the zoom in image on the right and with a scale bar that is burned in on the image that available in the software as well. So this is what we say publishable images of your fluorescence labeled tumor. Next thing, as mentioned earlier, we can, because all these studies required measure the changes over the time, okay, as the tumor progression experiment. So there are most time point analysis. So the mice are previously described experiment. We image a 3 time point, 4 days, 11 days. And also we have one that actually at 14 days, there's one mice that we image at 14 days. Okay. So in vivo, and the 14 days in ex vivo, we showed even here. Okay, so you can see the tumor actually become very visible after, actually not, it's on the day 11 images and also the day 14 images. It's more defined in that sense. So the scale bar is, as I mentioned, you can burn in, so allowing you to actually do a comparison, okay, in a very representative image manner, okay, across the time point. Okay, of course, the result of measurement is available here, but this is more about giving or generating a representative image that people can see at a glance on a manuscript or your presentation. Okay, so this GIF file, there are three red fluorescent protein positive mice and two negative control mice that is generated in Sapphire FL. They are captured at 100 micron resolutions and we scan at 0 to 3 focal plane. So that's why we can generate such a GIF file showing you the depth and also perhaps the shapes and also the signals of the tumor at that region. Okay, so it's clearly visible that we can see the red fluorescence protein, okay, over here, especially here, okay, that we are very clearly visible in the tumor, okay, but not at in the negative control because they have tumor, but they don't have any red fluorescent protein reporter, right? Okay, so this is ex vivo dissected tumor imaging. Okay, so the reason why we have ex vivo is to tally, as I mentioned, we need to tally the result that we image in vivo. Okay, so 4 of the mice were sacrificed. Okay, then we dissected the tumor and imaged directly, we put directly on the glass bed of the Sapphire FL. This is on day 11. Okay, and you can see the comparison of the signal on the RFP plus tumor versus the negative control tumor. Okay, so this uh, can be served as an important confirmatory steps, okay, in vivo studies because you have your negative control to show that yes, there is a tumor, but there is no red fluorescence protein. Okay, so that might be actually changes. You can utilize this in other kind of control, for example, like a treatment control, wild type control, and so forth. Okay, for your experimental design. Below is a GIF image of the tumors that image that different focal planes. Okay, so this is one focal plane, a single image, and this is a stacks of different images at different focal plane. We transform them into a GIF file. All right, so you can see the strong signals of the red fluorescence protein. We put it in this luminescence format, okay, and to showcase you the signal is very strong throughout the depths of the entire tumor. And but in contrast, if you look at the negative control, they only show slight autophoros near the edges. Okay, right. So let us look at another example of imaging of living small animal. So this is a PE-Sci5 labeled mouse tumor. Okay, so this tumor is huge. Okay, but it's internally, okay, internally huge. Okay, it's scanned at 200 micron resolutions and focus setting set at 1.55 mm. Okay, so this is as I mentioned before, this is the PE Psi 5, okay? The excitation and emission is actually is not the standard Psi 5. So the excitation is around Psi 3, but the emission is around Psi 5. So we able to image it by switching out the filters, okay? And put in this filter itself and image the PE Psi 5 signals. Right. So this is small animals on, I believe this is a black six mice. Right. So next thing we will look at is a zebrafish indeed. Okay. So this is Alexa Flow 488. Okay. And near infrared 800 labeled zebrafish. So you can see the channels listed at the lower left corner. Okay, it scans at 5 micron resolutions because it's a very tiny samples on the 96 well plate. So they need the resolutions to look at the signals in individuals zebrafish. As you look at the focus setting here is actually customized at 2 mm, okay, to look at the signals on the zebrafish itself. Okay, right. So contrast with the mice that we see before. Okay, so you can see that why in small animal, okay, that you don't really require the resolutions. You can always scan at higher, not higher, the higher look scan at 100, 200 micron resolutions. So in this case, you'll be able to save your time a lot and scan everything in a shorter time. But if you need to scan a living organism at a very high resolution, like 10 micron or 5 micron resolutions, then this will take slightly longer time, okay? Because you need the resolution to look at the individual signals on the samples. Okay, right. So that's the differences in terms of imaging living organism. We have also publications uh, previously, okay, on the Sapphire biomolecular imager. Okay, so here's are some examples that I put out from our publication notebook or book application book. So this is organ imaging. Okay, can see organ imaging on the using doing on the heart, looking at the oxidative stress, and this is another tumor studies. They ex vivo scanning the tumor, okay, and they're looking at the uh, the way that how they vaccinate and how it reacts in terms of the tumor progressions. And next thing will be the utilizing the autofluorescence signals from the organ itself. Okay, so this is harvested organ. Okay, then they're looking at the for autofluorescence signal and actually able to examine the abnormalities in the organ that they observe here, which is the liver. And of course, the observation of blood-brain barrier permeability also allows available to be done on Sapphire as well. Okay, so if you're interested, all these are available on our website on under the Sapphire publication note. Yeah. So let us look at Sapphire FL again. Okay, we have talked about how it will allow us to do a benchtop in vivo animal imaging, small animal imaging, your mouse model, your rodent, okay, or cover to zebrafish, C. elegans, and so on. Okay, the reason being is it has this wide field of view, allowing you to scan many small animals, including, like say, mice, rats, C. elegans, and zebrafish, and so on. Large, very large gel and phosphor screen for phosphor imaging. And with that, you have the option to decide what kind of resolutions that you want to go for. So Sapphire FL is not a Microsoft, it's a microscope itself, okay? But it is very good in taking high resolution images up to 5 micron resolution. So this is an image, okay, or in this example is a mouse lung tissue slide that probe with two target proteins and image on Sapphire FL at 5 micron resolutions. So you can see it provides detailed look at the structure of the lung tissue and also the distribution of the target within the tissue section itself. And the one that actually relevant to in vivo imaging will be the agaseous anesthesia system that we built in within the Sapphire FL. And this is compatible with third-party anesthesia systems. So if you already been doing that, it will just a matter of tubing and connection. Then you will be able to have the tubing like exhaust and everything, allowing you to scan your living organisms or living animal on the Sapphire FL. Signal wise, it uses uncoupled optical module design, okay, where it supports excitation and emission from UV range up to near infrared range. You can see these 12, okay, optical modules, standard optical modules we have in Sapphire FL, okay. So the pairing between the laser and filters, okay, you can pair differently. So because of the filter design, a lot it's like a USB. You can plug in, okay, use it or plug out and swap out. So these allow us to do over 30 unique combinations of the laser and filters that were custom to most of the dye available on the market, even including dyes that are newly discovered or a newly manufactured R&D for better signal or better signal to noise ratio, that kind of sense. Okay, right. So one of the best thing that come out from this uncoupled optical module design is we have this DAPI, not to say DAPI, but we call it 375 standard optical module, allowing you to image DAPI signals. Okay, so we Sapphire Alpha is the only laser scanning system that has this cover the UV range, allowing you to capture DAPI signals, okay, on that labeled on the cells, on the cell nucleus and nuclear stains. So there's a comparison on the in-cell Western HeLa cell lines, okay, staying with the DAPI, but we know usually couldn't get the signal under the in the 488 channels, but with the 375, sorry, is a standard optical module, we're able to get the signals, all right? And the uncoupled optical module design allow us to actually look at or utilizing a mega stock dies, okay? So the mega stock dies, they are basically have a huge excitation and emission spectra. We call it Stokes shift. Okay, the Stokes shifts are normally more than 80. Okay, okay, because normally the Stokes shift for the standard die like you see on the market like Psi 2, Psi 3, Psi 5, they are covered between like 10 to 40 the Stokes shift. But mega Stokes dies because they are a huge stokes between the excitation and emission wavelengths, the overlapping of the excitation and emission become very minimum. Okay, so it what it translates is it minimize the crosstalk between your excitation light and also the detection of the emission fluorescent signals during imaging, giving you a way better signal to noise ratio and better images as well. So with these, because they are new dies, they are not the standard dies that we have it on the market for years and years. Okay, so with the uncoupled optical module in the Sapphire FL, we're able to take out the on the 488 optical module. We just need to swap out the filter, and put in the 624 filter, and then we're able to image these megastock dies reliably. Okay. Right. So all these fluorescent signals, the one they either scan on your in vivo animals, organism, okay, all the plates and tissue slides and everything, they are supported with extended dynamic range. So what it means is you'll now be able to scan very high signal intensity, okay, samples and low signal intensity samples. For example, a knockout samples versus overexpressed samples with your wild type as your control at the same time without worrying about the saturation. Because if there is a saturation in the signal, okay, that even though it's generate a 16-bit file data, okay, the saturation that means you cannot quantify or do a data analysis on this image. But with extended dynamic range, since all of the pixels are not as saturated, you can detect or analyze these data or quantify the data without worrying all these saturations or signal linearity. So this is available and already built in the software. So if you're looking at the scenario, this is just example of if you have a fluorescent Western blot without the EDR function, okay, so there are bands that are usually saturated, okay, due to circumstances that you cannot lower the protein load anymore and so on. There are many restrictions on that. So, but with the EDR, you do not need to worry so much. Basically, all these will be captured in the extended dynamic range, covering the very low signals and very intense signals together without saturation. And this image, you can analyze it. Okay, that's the most important feature of this extended dynamic range. Okay. And as I mentioned before, so we have a confocal imaging technology using PMT and APD detectors in the Sapphire FL. Okay. So what it does is it has this um anything that outside the focal plane above and below will be filtered out. It will not be taken and detected. So it eliminates a lot of background or even unnecessary signals as well. Okay, so but we can set focal plane at different from -1 up to 6 mm, allowing us to actually image multiple array of a wide array of the application like your 96 well plate, 284 well plate, membrane, glass slide, gel, whole organ, and including the live animals or organisms. Okay, so here is a very uh, also comprehensive table on the applications that possibility that you can do in the Sapphire FL. So most importantly about this is this it must be a fluorescent range signals, okay, fluorescent range signals including the UV range and also it phosphor imaging as well. So that's the most important thing. And the second thing will be the resolutions that you want to go for and the depths of the signal you want to look at, okay. Right. So in anything that is lesser than the membrane or gel or plate, they will always excel in that. Okay, so here's some example. We have fourplex fluorescent spectrum plot, okay, fluorescent gels imaging, Comesi blue gels imaging, SDS page. Then you can image by 685 or near infrared 700 channels. Okay, PI, uh, PI assay to calculate the dead and dying cells versus the live cells assay and also looking at the gene transfection efficiency just by quantifying the yellow fluorescence protein, okay, on the wells itself. So look at this. This is basically an image, scanning image of the entire 24 well play. Okay, so it usually scan at 100 micron resolutions. That takes only 7 minutes. Okay, right. So that's why Sapphire FL is a very good benchtop imaging solution for your routine work that involve fluorescent signals. Tissue array. So we're moving on to higher resolution right now, 5 micron resolution because or 10 micron resolution because the size of the sample is basically very very small. This is only 1.5 mm in diameters. Microarray of formatted slides and also in-cell westerns, which is we actually talked about this in the previous webinar, uh, in depth about the in-cell westerns. So why we do that? Okay, so basically they are basically in adherent cells, allowing you to have multiple generate data points or generate data in high throughput manners. Okay, and comes along with it, we have the Azu Cyto in-cell Western kit that is actually a one-stop solution, okay, for all your in-cell Western protocol. All right, and then these are the data that can be generated from the in-cell Azu Cyto in-cell Western kit. So it's a triplex, okay, three multiplex fluorescence scanning on your in-cell Westerns, covering two, which is uh 700 and 800 channels, and the sign 3 channel for your what is that called? We call it total cell stains, yes, for your normalization control. Other than that, Sapphire FL also provides a solution for your phosphor imaging. So it uses a PMT, a detector for phosphor imaging. Basically, it's an assay that has a higher sensitivity because of radio labeled samples. And then we use a phosphor screen to capture the signal from your sample and then image the screen afterwards. So definitely applies for carbon 14, hydrogen free, phosphorus 32, and sulfur 35, or so on and so on about maybe about nuclear study. So there are involving like tagging several uh, components such as protein lipid, glycoprotein, saccharide, and so on and so forth, or nucleic acid. Okay, so this is just showing you the radioactive samples, how we image it. This is the chemiluminescence module that I mentioned before. If you want to have able to image chemiluminescence, there will be an additional or optional module, okay, allowing you to capture chemiluminescence signals. And this chemiluminescence module allowing you to do densitometry analysis as well, okay, based on on the optical density that you can do or quantify and generate standard curve to have a standard or QC for your, let's say, Carmesy blue or silver stains, protein gel, and X-ray films images. So here's her Sapphire FL biomolecular imager, where we talk about the in vivo animal imaging before on red fluorescence protein positive tumors. And then we have a lot more publication and application notes that we released just two few weeks ago. Okay, so one notable one that we utilize the fluorescence assay is biphase assay. You can see is actually looking at the protein ligand interactions and also in-cell westerns, okay, and then microarray scanning and also autophagy associated assay. They're looking at the fluorescence signals of the autophagy markers. So all these are available, including the publication booklets, okay, that are available on our website, the publication booklet, and also all the application notes. Feel free to look at them and see how we use Sapphire FL to get all these publishable data. Okay, right. So in summary, Sapphire FL is a very useful tool. It offers you a benchtop solution for your basic fluorescence signal capturing at a very high sensitivity and resolutions. Okay, so it's a very advanced imaging, and that we extended it into in vivo animal imaging, including small rodent like mice, okay, and very skinny rat, and also zebrafish, okay, C. elegans, and all the other organisms that you can think about that have fluorescence tag, okay? And the one that actually dedicated purposely for small animal imaging is the 5 gaseous anesthesia ports that is built in on Sapphire FL that is facilitate you in terms of scanning the animal in vivo without going through a bulky instruments and also without going through all the troubles. Okay, so it has been well received all over the world. All right. Okay. So most of them are looking at the high resolution fluorescence scanning, and some of them in the nuclear research, for example, the one in Denmark and here and the cancer research, they're utilizing the phosphor imaging, okay, the module for their radio labeled samples. So here's Azuba Systems. We have imaging solution for your basic and lab applications. All right. So that is the Sapphire FL Biomolecular Imager. I am Kylie. Thank you for your attention and time here. So if you have any inquiry, feel free to contact us or contact Korea or Ban over here. Okay. Right. Thank you so much. I'll pass the floor back to Ban then. If you have any question, please feel free to type in the chat. Thank you.